Mustafa Diken is also coming from Mainz, but he's not here. He cannot be here, but he's online, so great, Mustafa, to have you here. He is working for TRON, the Translational Oncology branch, so to speak, of BioNTech, and so he can do still a lot of scientific basic questions. He's joining us today with his title, Employing mRNA Against Cancer. Mustafa, the screen is yours.
Okay. Hello, everyone. Sorry that I couldn't be here. Today, I would like to give you some examples of how we make use of RNA against cancer. So these are my disclosures. Here, this is the mRNA technology that we are using. We formulate RNA with, in this presentation, I will show examples of RNA lipoplexes that can be used to immunize animals or human, and there the RNA is internalized by endocytosis and then escapes from the ribosomes, escapes from these endosomes and gets translated by the ribosome. And then we have our protein translated just like any other RNA in the cell. Then this protein is either can be secreted or can be processed by the MHC class 1 processing and can be re-internalized and brought into the vesicles into which have MHC class 2. And at the end of the day, you can have MHC class 1 and MHC class 2 presentation of the RNA encoded antigen to induce immune responses. So we are producing RNA by using a DNA template that can be simply generated by any genetic engineering methodology and using an in vitro transcription cell-free methodology to have a single-stranded, non-modified uridine-containing RNA with different elements like the 5' cap, 5' and 3' UTRs, and certain regions to improve the class 1 and class 2 presentation, and also poly A. And then we formulate and then bring it into action. So RNA has a dual mode of action. First, it delivers the information for the generation of the antigens, so for the ribosomes. And it also has an interesting adjuvanticity by signaling through innate immune sensors to induce innate immune responses. So by this, you don't need an extra adjuvant for your vaccine. So it is there's no protein, no haplotype restriction. It can be optimized, as I mentioned, by any type of molecular engineering. It is safe, and it has been shown safe in many different clinical trials, we all know. And manufacturing is very versatile. It can be manufactured at very high amounts that we can produce billions of doses versus in very small amounts for example, personalized cancer vaccines. So talk about the vaccines. So we have two platforms called FixVac and iNeST. FixVac is the off-the-shelf mRNA immunotherapy targeting shared antigens, tumor-associated antigens of the cancer. Then we can combine different antigens and immunize patients. And the iNeST approach, it's individualized neoantigen-specific immunotherapy. We map mutations in the tumors, predict neoantigens, and really manufacture RNA on demand for each and every patient, and then and have an individualized immunotherapy. We can go up to 20 neoantigens and treat every individual patient across many different solid tumor types. So here for this we are using, as I mentioned, RNA lipoplexes. And what are mRNA lipoplexes? So these are nanoparticles in 200, 300 nanometers in diameter, and we formed them by complexing RNA with cationic liposomes. And here, when when you immunize the mice with luciferase encoding RNA in a naked format intravenously, you don't get any luciferase signal. But when you complex it with these cationic liposomes at different ratios, you have an interesting biodistribution. When you go more for the negative charge, you go to the spleen, and when you go for a more positive net charge, your biodistribution of the RNA encoded protein is more in the lungs. And it's not only spleen in this negative charge molecule, it's also the lymph nodes and bone marrow, which are the main immunological organs. So we can bring the RNA into immunological organs and also protect RNA from degradation. We looked a little bit at the uptake and pharmacodynamics of the RNA. So when we inject the luciferase encoding RNA lipoplexes, and then deplete dendritic cells in mice, we see a significant reduction in the luciferase signal. And when we employ an EGFP encoding mRNA, we can nicely see that it's internalized mainly by dendritic cells and macrophages in the spleen and other lymphoid organs. And as I mentioned, we have also an activation of the lymphoid organ cells. And here you can see that the dendritic cells upregulate their maturation markers thanks to the intrinsic adjuvancy of the RNA, as well as we have activation of the NK cells and B cells. So you might wonder, why are they getting also activated, although they didn't internalize RNA? Here we have a nice mediator. It's interferon alpha. And thanks to the adjuvant activity, we have systemic interferon alpha, which is really systemic, and these cells also have interferon alpha receptors, and this in turn activates these cells as well. Of note, interferon alpha is itself an anti-cancer drug used in the clinic. So we looked as vaccine developers whether we can induce T cells, antigen-specific T cells. We employed model antigens like GP70, and by repetitive immunizations, it's a very potent platform. You can reach up to 50% of the whole CD8 T cells can be antigen-specific. These T cells can kill target cells that are antigen loaded in an in vivo cytotoxic assay. We can get really functional T cells that are secreting interferon gamma and granzyme B. But interestingly, when we block interferon alpha signaling during the vaccination, these T cells that we generate do not secrete anymore such molecules, effector molecules. And we also have very good memory responses that are very important for the cancer field. So we use this also in some tumor experiments, and you can nicely see that in a colon carcinoma model, when we immunize with mRNA coding our lipoplexes, we have total free of tumors in the lungs of the mice. This is another example. This time the luciferase is coming from the tumor cells. They are growing happily in the lungs, but when we immunize, the growth is completely inhibited. Another model, B16 melanoma, using a tumor-associated antigen, TRP1. Immunization with this antigen leads to the shrinkage of the tumors and loss of the luciferase signal. And when we use an irrelevant RNA that is not expressed by the tumor, we don't have any effect. These are again the luciferase pictures. They are completely free when they immunize. And of note, all these experiments are therapeutic tumor experiments. We first inoculate the tumor and then come up with the RNA. And these are the lungs of the mice that are free of the tumor nodules. Here's a subcutaneous tumor model, TC1 head and neck cancer model. When the control mice all grow, and when we use an irrelevant RNA, that's also growing. All the mice and tumors are growing. When we employ an E6, E7 encoding RNA, these are viral antigens expressed by this tumor. And we have shrinkage of the tumors. And you can see when we start the therapy on day 7 or day 10. And these are some pictures that showing the shrinkage of the tumors. And there's a complete tumor control and survival of the mice with this type of immunization. What we observed in this TC1 tumor model is that the tumor cells start upregulating PD-L1, an immune checkpoint molecule expressed on the tumor cells that is blocking the T cell functions. And it's really happening, especially when we use an antigen-specific vaccination. And we asked the question whether we can then combine the PD-L1 antibody with our vaccination. PD-L1 is, as you know, as a checkpoint inhibitor antibody used in the clinic and marketed. And this is a very good combination. And here we are using just a single vaccination combined with the PD-L1 therapy. It works very well. And it also works very nicely with other type of standard of care, like such as radiotherapy. Here you can nicely see that when they are used alone, each therapy is mildly effective, but when we combine both of them, it's really effective against the growing tumors. So with all this data, we started the first in human clinical trials in melanoma patients using 4 different shared antigens and immunize the patients with RNA lipoplexes. Here we can see that there's also metabolic activity in spleen shortly after vaccine administration, as we observed in mice. And similarly, we also have interferon alpha as a pulsatile induction, and it's transient and it is dose dependent. We can induce T cell responses in many patients against the antigens. Some of the responses were not existing, and then we induce them after the vaccination. Some of them were pre-existing, and we can further enhance the response. These are some clinical data. It's also published. You can look at the details that we can see shrinkage of the tumors. We can again see multiple responses. And of note, when we isolate these T cells that are primed after and generated after vaccination, and then co-incubate them with the tumor cells derived from the same patient. We can see very nice killing ex vivo, and they are also showing still antigen specificity. So the other part that I taught you was the neoantigen part. Here we can nicely make use of the neoantigens. What are neoantigens? They are the changes in the DNA. They can be point mutations, deletions, insertions, splice site mutations, or gene fusions that can be employed to generate an antigen. So what we did in the past is that we sequence the tumors and healthy tissue, compare, find the mutations, prioritize them, make vaccines, and then test it in the mice. And here we are doing very similar thing in the human setting as well. This is the mouse data, just to show shortly that we can use identified mutation 30 as a neoantigen, and vaccination with this led to loss of the tumor, so tumor control, and two different models, B16 and CD26. And when we look different models, so we can have really both MHC class 1 and class 2 restricted neoantigens that can be employed for vaccination. With this, we also started the clinical trial in which we immunized my patients, again, melanoma patients with 10 different neoantigens, this time without any nanoparticles or naked RNA injection. We can induce T-cell responses in all of the patients. It differed from patient to patient, against how many antigens we have the immune response, but it was really successful. And just to show you that these patients experienced the multiple relapses before the mutalum vaccine, our individualized vaccine, but after the vaccine, it was significantly reduced, as you can see in the graph. And this study was really done or started in around 2013-15, and now we have also quite some follow-up of the patients for 4 or 5 years. And you can nicely see that even after we stop the vaccination here, we can still see T-cell responses that can be seen by multimer stainings. And interestingly, these T-cells, when they are taken out from the patient and then co-incubated with the tumor cells, they can still kill the tumor cells. So they are really still very active. So last part, I just want to touch shortly on a common problem. So we can generate vaccine, we can generate very efficient T cells, but what we observe also in our clinical trials is that the tumors tend to either downregulate or delete the beta-2 microglobulin, which is an essential part of the MHC class 1 complex. So without this, there is no stable class 1 and no antigen is presented. So no matter whether you have very good T cells, they cannot find and kill the cancer cells. What are we doing for this? And this is also what we showed. We generated some beta-2M knockout mouse models to mimic this. And interesting, also the checkpoint inhibitors are not really working very well in these models. So what we did is we combined a monoclonal antibody approach that is against the tumor antigen versus an mRNA encoded IL-2 that is formulated in lipid nanoparticles for liver targeting and have high systemic availability. So when we combine these, you can nicely see that the combination of them works very nicely in this beta-2M knockout mice in 2 different models, B16 and MC38, using antigens against them. And also interestingly here, addition of these 2 to the immune checkpoint blocker circumvents positive selection of MHC class 1 deficient cancer cell clones. That means that also the MHC class 1 deficient cancer cells are killed when we add these complex to the checkpoint inhibitor therapy. And here I tried to identify the crucial effector cells. AK cell depletion did not have a real effect, but especially the CD8 T cell depletion and interferon alpha neutralization has a big effect on the therapy success, as well as the macrophage depletion. So we believe that this therapy efficiency is dependent on the macrophages, CD8 T cells, and interferon gamma. So what happens is that in the MHC class 1 positive tumor, everything is hot. Then we have response to the immune checkpoint blockage, chemo and radiotherapy, because we can have the T cells infiltration, macrophage infiltration. When we have the deficient tumor, it's a dessert, so that we don't have any tumor cells. Everyone expected we have a lot of NK cells, but it's not. And what we do is with the IL-2 treatment that is delivered via LNPs of IL-2 RNA to the liver, then it goes to the tumor and attracts the CD8 T cells, expands them, and they come. But you also need the macrophage component to present the antigens, because the T cells cannot see the antigens on the tumor cell due to the beta-2M knockout, and the macrophages enable also to detect the antigen thanks to the antibody we use, and then with this complex we again restore the hot tumor microenvironment and the therapeutic responsiveness. And we are also trying to employ this into the next setting together with our vaccines and both cancer vaccines that are made up of shared tumor antigens, but also individualized neoantigens. So with that said, I would like to thank you everyone and acknowledge the people involved.
Thank you, Mustafa. And thank you for showing us that we can do more than vaccination against viruses, but that it can be employed also against tumors. And yeah, I think that was the initial idea of BioNTech and of Ugo Shahin and Özlem Türeci, and also, of course, with Mustafa. He has been there for, I think, since the start of all of that.